Our speaker this week is one of our CCMB affiliate faculty, Sriram Chandrasekaran, and he's assistant professor in the Department of Biomedical Engineering. And let me just give you a quick overview of his bio here. He has his bachelor's degree in biotechnology from Anna University, and he completed his PhD in biophysics at the University of Illinois. And after finishing his PhD, he worked at Harvard and MIT as a Harvard Junior Fellow, and then came to Michigan in 2017. His lab develops systems biology algorithms for drug discovery, and he's going to tell us more about that today. And his research has been published in outstanding journals like Cell, Genome Biology, and PNAS. And he's received a number of prestigious awards, including the Harvard Junior Fellowship, the Howard Hughes Medical Institute International Predoctoral Fellowship, the 2014 William Milton Fund Award, and a 2018 Precision Health Investigator Award. So without further ado, I'll turn it over to you, Sriram, and we're looking forward to your talk.
Thanks, Josh. Thanks for the introduction and inviting me. So, as Josh mentioned, my lab works on two main areas, and today I'm going to primarily talk about new methods my lab has developed for predicting drug combinations using machine learning and AI. So overall, there are two main themes my lab is working on. One is using machine learning and AI for drug discovery. Specifically, we are focusing more on infectious disease and trying to design new treatments that take into account the properties of the pathogen, its metabolism, its environment, its interaction with the immune system, and how we can design more personalized therapies by taking a systems approach that integrates all these data sets and coming up with more personalized precision medications. Another area that my lab is working on, which I won't have time to talk about today, is developing new systems biology methods for studying metabolism and regulation. So we've developed a slew of methods that take into account transcriptomics, proteomics, epigenomics, and metabolomics data, and use that to understand and predict metabolic behavior of various cell types, microbes, stem cells, cancer cells. So we can definitely talk offline if you're interested in these methods. So today I'm going to focus more on our drug discovery research. Specifically, one main problem I'm going to talk about is antibiotic resistance. As you probably have heard news, antibiotic resistance is a huge problem right now. And one main issue is that many of the antibiotics that you're using now are slowly becoming ineffective. Antibiotics are one of the most, I would say, greatest inventions of mankind. Before antibiotics were invented, even a simple wound or a scratch can become life-threatening. Like previously, before antibiotics, 1 out of 10 simple infections results in loss of limb or even life. Similarly, 3 out of 10 people who had a pneumonia infection died. So, so antibiotics have had a huge impact on our society. But due to drug resistance, we are sort of going back to this grim scenario where even a simple wound can become life-threatening. So why did we get here? So one major reason is just plain evolution. So, this is an example for one infectious disease, gonorrhea, where, the y-axis shows the year antibiotics were introduced against this disease, and the x-axis shows when resistance was reported. And you can see that sulfonamides were introduced in 1935, and within 5 years, a resistant strain was reported. Similarly, new drugs were introduced in 1940s, 1950s, and within a few years, people found drug-resistant strains. And so this would have been okay if you keep discovering new antibiotics, but unfortunately, that's not the case. So you can see from this graph here that, in the 1950s and 60s, there were dozens of antibiotics that were discovered and introduced by FDA. But then as you move towards the 1990s and 2000s, there's hardly been any new antibiotics that were approved by FDA. So that's the main problem. There's a combination of lack of new antibiotics and increasing resistance that's causing this pandemic. So then you start hearing news like these where, for example, New York Times a few years back reported that there's a strain of E. coli that is resistant to all known antibiotics. And people have reported similar strains for, like Staph aureus, MRSA, Acinetobacter, where now these pathogens are now resistant to all known antibiotics. So you just have to rely on your immune system to, survive these infections. And unfortunately, all these are being caused by our use and abuse of antibiotics. And so my talk is going to focus on how we're going to address this challenge and how, like big data and machine learning can help ameliorate this crisis. So one major solution for tackling antibiotic resistance is by using what's called as drug combinations or combination therapies. So drug combinations have a lot of promising properties that can help with this crisis. One is that the probability of resistance occurring when you have multiple drugs is lower, just sort of statistical chance, than when you have just a single drug. So by using combinations of 3 or 4 antibiotics, you lower the chance of resistance occurring in the first place. Another useful property is that you could enhance the potency of antibiotics or other drugs by using them in combinations. And third one is the spectrum. That is, by using combinations, you can target different subpopulations of bacteria, some that are slow growing, fast growing, or with different genotypes. So you can broaden the spectrum of cells that are being treated. And the fourth one is that you can potentially repurpose drugs using this. For example, some drugs might have low potency on their own, but then in combinations, they might work really well. So there are a lot of great reasons for using drugs in combination, but there are challenges too when you're combining drugs. One common challenge that people always mention is that it's the combinatorial complexity. So when you want to pick, let's say, just 4 drugs from a combination of 100 potential candidates, the number of combinations explodes exponentially. For example, in this case, choosing 4 drugs like we commonly used for treating tuberculosis from a set of just 100 would require screening close to 4 million combinations. And now if you also want to include the dosing, for example, if you have a low dose and a high dose, then combinations become like, tens or hundreds of millions. And so that's one huge challenge when designing combination therapies. But there are many others too, which I'll talk about today. So the first one is the large number of combinations. But the other is that even if you can use AI or machine learning to automate this process, there are, other challenges. Like, for example, for many organisms like tuberculosis or Staph aureus, we don't actually have enough data to even start building these machine learning models. And so I'll talk about how we can address this challenge. And the third challenge I'll talk about is how the metabolism of the pathogen influences its response to antibiotics. So one thing we have discovered and others have found is that when you change the metabolism of the pathogen, either if it's growing in hypoxic conditions or in starvation conditions, its response to antibiotics changes dramatically. And this also occurs in cancer cells, which I'll briefly mention in this talk. And then another main problem is that the conditions in the lab in which drug discovery is done is obviously very different than the conditions inside the body, in the lung or gut where the infection is happening. And this causes both biophysical problems, like the antibiotics might not penetrate or reach the site of infection. And also it also changes the environment in which the bacteria is growing in, so causing other issues. So these are the 4 main challenges that we're talking about today and how we can use machine learning to address these. So let me start the first problem, which is the large number of combinations. To address this, we developed a method called INDIGO that uses machine learning to sort of screen through these large number of combinations. And the way INDIGO works, sort of how the Netflix movie recommendation algorithm works. In Netflix, you provide uses 2 inputs sort of to recommend movies. The one is known examples of movies that you've watched previously, right? That's one input. The second input is like a database or properties of movies. For example, is a movie a comedy or is it, like horror? And also, what were the characters involved in the movie? So, depending on all these properties, then it connects that with your previous movie watching history to recommend new movies or TV shows for you. So, that's sort of how Indigo works. It takes two inputs. One is known examples of drugs that are known to be synergistic or antagonistic, and then the properties of these drugs. In this case, we're using a data set called chemogenomics to understand the underlying properties of the drugs. So, what is chemogenomics? So, in chemogenomics, what you do is you try to identify uh, genes or pathways that are involved in response to a drug. So, a simple chemogenomic screen will involve, taking a microbial strain, deleting uh, various genes in the genome, and then exposing those to a drug of interest. And using this data, you can identify genes that, when deleted, can cause resistance to a drug or increase the sensitivity to a drug. So we call these drug gene interactions or chemical genetic interactions. So this is a very rich source of information. It not only tells you what the actual target of the drug is, but also other processes that are involved in drug action, like what proteins are involved in efflux of the drug or entry of the drug into the cell or its metabolism inside the cell. So it sort of gives you the entire set of processes that are involved for a drug to act against a cell. So this is the data that Indigo uses to infer drug properties, and it then ties this with data on known drug-drug interactions. So this is like the overall mechanism behind how Indigo works, is that it takes drug gene interactions that it learns from chemogenomics data and then builds sort of a drug gene interaction network and then uses that information to predict drug-drug interactions. So now this can be scaled to multi-drug interactions as well. So the way it connects these two sources of information is using a machine learning algorithm called random forest. So as I said, we need two inputs. One is the known data on how drugs interact, which I'm showing in the bottom. For example, drug 2 and drug 1 could be synergistic, drug 2 and 3 are antagonistic. So this is like known examples of drug-drug interactions that Indigo uses. Another is like the chemogenomics barcode for each drug, like what genes cause resistance or sensitivity to a drug of interest. So with these two data, it can then connect genes in the chemogenomics profile that are predictive of synergy or antagonism. So that's shown by the decision trees here. Random forest algorithm builds a series of decision trees that connects the chemogenomic data with the ultimate outcome, whether two drugs are synergistic or antagonistic. Once this model is built, you can now use this for any drug for which you have chemogenomics data. So in this case, if you have novel drugs like drug 6, drug 7, Indigo can now predict how these novel drugs interact with each other. So we first built this model for E. coli, and for building this, we generated drug interaction data for a set of 15 different antibiotics. And that's what's shown here in this triangle. There you can see a bunch of drugs, and red means that they are antagonistic, and blue means they're synergistic, and white means they are linear. And we usually test these drugs in multiple doses. And so that's what's shown in gray boxes inside that tell you what the growth of the bacteria is in different doses. So you can see in cases when they're antagonistic, there's a lot of growth of the bacteria in multiple doses. When they're synergistic, there's less growth in different doses. So this is the input that goes in to Indigo. These are known examples of synergistic and antagonistic interactions. And then we link this with chemogenomics data for each of these drugs. And in this case, we used a compendium of over 324 chemogenomic profiles of various drugs, stress agents, and other metabolites. And so using this now, we are able to predict. We start with all pairwise combinations of this data, So by learning patterns from this, Indigo was able to predict over 50,000 combinations of various drugs. So that's the prediction. So then we decided to test these. So to test this model, uh, we picked highly synergistic or highly antagonistic combinations. And then we picked drugs with new mechanism of action that was not part of the training data. So we picked 4 drugs that were very, very different from the ones that were used in training. So this sort of gives you like a worst case scenario for Indigo. You see a completely novel drug, how, and then you can evaluate how good its predictive power is in this situation. So we then experimentally measured the drug interactions for these novel drug combinations. And what you can see here is that once again, Indigo was able to accurately predict uh, synergistic and antagonistic interactions. So the table here, we were able to pick like the top 10 synergistic ones. 6 out of 10 were confirmed experimentally. Similarly, 7 out of the top 10 antagonistic predictions were confirmed experimentally. Overall, there was a very good correlation between what the model predicted and what was, experimentally observed. So there was a strong, significant difference between synergistic predictions and antagonistic predictions. So we've also been able to do this for higher order combinations. I'm showing an example where we experimentally tested 3-way combinations, once again with new drugs, like 52 combinations involved, completely novel drugs not seen by Indigo. And one thing you'll notice here is that red is antagonistic and green is synergy, is that as we go up higher to higher order combinations, synergy becomes more and more rare. We think that antagonism dominates uh, synergy, and so when you have, for example, 2 drugs that are synergistic, and then you add an antagonistic drug on top of it, the resulting outcome becomes more antagonistic, and so synergy is really rare in higher order combinations. That means you definitely need tools like Indigo to search through millions of combinations to find those that are highly synergistic. And even in 3-way combinations, we had a very strong correlation, almost the same accuracy as we found in the pairwise combination data. So far I've told you that Indigo seems to be very effective in predicting drug interactions, even for completely novel drugs. So I want to briefly jump into how Indigo is able to predict this. So we decided to focus more on the underlying genes used by Indigo to predict drug interaction. So I just want to briefly talk about what genes were used by the random forest algorithm for predicting drug interactions. So one thing that we found surprisingly was that even though the chemogenomics data has several thousand genes, uh, Indigo was using just the top 250 genes were sufficient to account for 75% of the model's predictive ability. So only a small set of genes were being used to make predictions. And then what we found is that when you looked at the pathways that were represented among these top 250 genes, many of them were connected to known targets of antibiotics, but there were also other surprising pathways that were not direct targets of antibiotics, like, for example, amino acid metabolism or oxidative phosphorylation. So these are not the direct targets, but surprisingly, they are uh, being picked up by Indigo as being important for drug interactions. So recent studies have shown that metabolic pathways seem to modulate drug interactions. So now I'll talk about, dive into these mechanisms in the second part of the talk. And so far I've told how we can explore the large number of combinations using Indigo in a model organism like E. coli. But then most of these treatments are urgently needed for pathogens like Staph aureus, which is a major pathogen in hospitals, or tuberculosis, which is a pathogen that occurs worldwide. But for these pathogens, we don't have such rich detailed data like we have for E. coli or uh, Saccharomyces cerevisiae. So for these organisms, we need like a modified approach, which I'll talk about here, where you can leverage data in model organisms like E. coli, but still apply it to pathogens like Staph aureus and Mycobacterium tuberculosis. So how do we make predictions for another species? So uh, as I said before, like this type of data sets are not available outside of E. coli. And so our idea was that maybe we can use the same data set that we've used for building an E. coli model, but then leverage it to apply to pathogens like Staph aureus by using the idea of like evolutionary conservation or orthology. So one thing we discovered was that many of the genes that Indigo was using, like the top 250, seem to be highly conserved uh, across evolution. So we thought maybe we can take the Indigo model for E. coli, find the genes that are conserved in an organism like Staph aureus, and then use like an orthology-based model to make predictions in another organism. So we'll take the same Indigo model, delete genes that are not conserved, keep the ones that are conserved, and then see how that changes the drug interaction predictions. So that's what we did. We first matched orthologs, that is genes that are conserved between E. coli and Staph aureus. And even though there were only 840 genes that were shared between the two, they were highly enriched among those that were in the top 250 genes that were considered to be important by Indigo for predicting drug interactions. So then we kept those conserved genes and deleted those that were not conserved, and then we recalculated the interaction scores. So overall, we expected that drug interactions will be conserved, given that these genes were conserved. But then we found that there were some interactions that were highly variable between the two organisms. So to test this, we once again experimentally measured uh, drug interactions for antibiotics that we previously measured in E. coli, and then we compared how this fared with the Indigo predictions. So what we found was that Indigo was surprisingly able to predict drug interactions with pretty good accuracy, even in Staph aureus. So I should mention that the correlation is not as high as E. coli, but then we are getting this data for free. We're using E. coli data to make predictions in a very different organism, Staph aureus, which is a gram-positive microbe, but you're still able to accurately predict drug combinations that are synergistic and differentiate them from those that are antagonistic, just based on like the evolutionary conservation of genes between the two microbes. So we also done this in other organisms. So another pathogen our lab is really interested in is Mycobacterium tuberculosis, and we applied the same idea in this pathogen, where we once again took the E. coli Indigo model, matched the orthologous genes between E. coli and Mycobacterium, and then we made predictions of drug interactions in Mycobacterium. And in this case, we just compared this with data from literature, and once again, we were able to accurately identify synergistic drug combinations from those that are neutral or antagonistic with high accuracy, even in this organism. So I should add that like Mycobacterium tuberculosis is of high interest to our lab. And one thing we noticed when we built this model is that many of the drugs that were being screened in E. coli are not the ones that are clinically relevant for tuberculosis. So then we thought maybe we need to build a more broader model for Mycobacterium. So for Mycobacterium tuberculosis is like a worldwide pathogen that kills around 1.4 million people annually every year, and treatments take more than 6 months, and there's a widespread drug resistance for this pathogen. And so we decided to use Indigo to identify multi-drug combinations against tuberculosis, and to increase our pool of drugs that we are looking at, we built a model specifically for this pathogen. The way we did this was we collaborated with David Sherman's lab at University of Washington, where we generated new data for this pathogen. We also compiled data from literature, and we made one more change to Indigo. So far, I've been talking about chemogenomics data for identifying drug-gene interactions. but it's extremely hard to generate this data for pathogens like mycobacterium. So we decided to use transcriptomics data instead. So in transcriptomics, you know, you don't need to do gene deletion. You just have to do RNA sequencing to get the cell's response to different drugs. And we infer drug gene interactions by identifying genes that either go up or go down significantly after drug treatment. And so, we compiled and generated new transcriptomics data for various drugs in this pathogen. And then we also compiled known drug-drug interactions in this pathogen. So using those two data sets, we then built an INDIGO model specific to tuberculosis. And then we made predictions for all possible 2 or 3 drug combinations against this pathogen. So since we have a much bigger model now, so when we made predictions for all pairwise and all three-way combinations, it came out to be around 5 million different combinations. So I'm just summarizing them here in these simple box plots. And one thing we found was that drug combinations that were currently being used clinically for treating tuberculosis tend to be on the more synergistic side, compared to most randomly picked drug combinations. You can see that here. But since we had like 5 million combinations, we also identified a few more combinations that were more synergistic than the ones that were being used clinically. And so we then decided to test those combinations experimentally to see how well they fared uh, compared to the predictions. So that's what I'm showing here in the bottom panel. We picked 32 combinations that were either highly synergistic or highly antagonistic predicted by the model. And then we compared those with experimentally measured drug interaction scores. And we found a very high correlation again between what the model predicted for both pairwise, shown in red, and three-way combinations, shown in blue here. And we also found highly surprising combinations. For example, one novel combination that we found was between the drugs spectinomycin and chlorpromazine. So I share that chlorpromazine is not a TB drug; it's actually a antipsychotic drug uh, that people are thinking of repurposing for infectious disease, specifically for tuberculosis. And what we found, Indigo predicted them to be highly synergistic, and what we found experimentally was also that it was highly synergistic. And you can see from this experimental assay here that when you add like 1x or 2x the normal MIC of spectromycin, then you get complete bacterial killing, shown here in red boxes. Uh, Similarly, when you add 1x or 2x of chlorpromazine, you get complete bacterial killing. But then when you combine both, when you just add 1/16 of chlorpromazine and 1/16 of spectromycin, you get similar killing. So, by combining them, you're able to reduce the dosing close to tenfold. So this is one of few surprising combinations, highly sensitive combinations that we found using Indigo. We've also been able to engineer synergy or even understand the mechanisms behind drug synergy using this approach. So, for example, we tried to identify genes or regulators that can influence drug synergy in these organisms. And so the way we could do that using Indigo is by deleting different genes and then calculating how they impact drug interactions. So we took the tuberculosis Indigo model and then we deleted groups of genes that are known to be regulated by transcription factors. And then we tried to identify transcription factors that could either enhance synergy or antagonism between different antibiotics. So what I'm showing here is our virtual screen where we screened over 200 different transcription factors and then tried to determine how they influence drug synergy and antagonism. And one transcription factor really popped out that had a huge impact on multiple drug combinations, which is an unknown gene called RV1353C. And then we then start to analyze how it influences drug interactions, between multiple combinations. And that's shown here in panel B, where, when we looked at multiple drug combinations, there were some combinations where it had a huge impact, shown here in the top, and some combinations had a minimal impact. So we decided to experimentally test how this transcription factor influence drug synergy between these antibiotics. And so we picked two extremes, for example, the bedaquiline streptomycin combination, uh, and then the chlorpromazine CFZ STM combination as a negative control. And what we found was that when you knocked out this transcription factor, uh, it made these combinations more antagonistic, as shown here on panel C. And if you overexpress it, it made them more synergistic. So, by using INDIGO now, you can rationally engineer synergy and antagonism. So, in contrast, for the negative control, you can see that deleting this transcription factor did not have a significant impact for the last drug combination. So using INDIGO now, you can sort of understand the mechanism behind how drug synergy works and also rationally engineer synergy between different antibiotics. So one more thing I want to add in this study is that we next asked, so far I've shown you data on in vitro where we can predict synergy or antagonism between drugs, but we don't know if this really matters clinically. Like, does drug synergistic combinations that you found in the lab, do they actually work better in clinical trials? So to test that, we compiled clinical trial data for various drug combinations to see how they, if synergistic combinations did better in the clinic. And so this is how the clinical trial data looks like. They tested different combinations in various patients, and then they calculate what's called as a percentage uh, negative culture. So that is the percentage of patients who show negative bacterial culture after 2 months of treatment. So you can see here that there are some drug combinations that are just about average, like only 50% of the patients show negative culture, and there are others that are close to 90%. So then we decided to compare the drug interaction scores for these combinations and compare them with the clinical performance. And to our surprise, we found that highly synergistic combinations do indeed do well in the clinic, uh, as shown in this plot here, where the lower INDIGO score means they're more synergistic, and you can see that uh, drug interactions with low drug combinations with low INDIGO score had a much higher percentage negative culture in the clinical trials. So this shows that drug synergy is a great way to identify promising drug combinations, and in general, drug synergy correlates very well with clinical efficacy. So I would say this is surprising because we've tested these in lab conditions, and there are many other factors that could influence clinical outcome, like toxicity or uh, side effects or development of uh, resistance. But in this case, we still find that in vitro drug synergy still is predictive of clinical efficacy across multiple clinical trials. So to summarize what I've said so far, I talked about the INDIGO approach and how we can use that to screen large number of drug combinations. And then I also talked about how we can use the evolutionary conservation of genes to predict drug interactions in uh, less studied pathogens like Staph aureus or Mycobacterium tuberculosis. And then I also showed how uh, synergistic drug combinations correlate well with clinical efficacy in clinical trials. And then we also showed how we can understand the mechanism of drug interactions and potentially use that to rationally engineer a highly synergistic drug combination. So for the next part of the talk, I'm going to talk about the other two challenges that I mentioned, which is that understanding the role of how metabolism affects drug synergy or drug efficacy, and how we can better capture the in vivo infection environment and use that to predict more precise drug therapies. For that, I just want to pause for a minute to see if anyone has any questions.
Sriram, Margaret here. When you have found an interaction on, let's say, tuberculosis mechanism between a known tuberculosis drug and clozapine or whatever it was, a anti-schizophrenia drug, would you predict the interaction would also work the other way around? So would you also predict that you might be able to reduce the dose of clozapine by adding an anti-tuberculosis drug to it? Or is the mechanism in schizophrenia so completely different that you can't take it the other way around?
So I would say that the predictions are still cell type specific. So, the predictions that for tuberculosis might change in a different organism or even a different strain of tuberculosis, but we can definitely predict that if the genes involved for these predictions are conserved, then I would expect the mechanisms might also be conserved. So if those genes produced by the model for chlorpromazine are also conserved in mammalian cells, then maybe the mechanism might also be conserved. All right, looks like there are no other questions. So I'll move on to the second half of the talk. There I'm going to talk about how, metabolism and the in vivo environment could affect these predictions. So one recent discovery that people have found is that metabolism seems to have a huge impact on the potency of antibiotics. Like traditionally, antibiotics target non-metabolic processes like transcription, translation, but surprisingly, their metabolism seems to have still a huge impact even on antibiotics that don't impact metabolism. For example, in this very interesting study, they took a drug-resistant bacteria that is resistant to an antibiotic called kanamycin, and then all they did was they added two metabolites, alanine and glucose. And surprisingly, by adding these two metabolites, drug-resistant bacteria became drug sensitive in this case. So even though the resistance is genetically mediated, by changing the metabolism of this drug-resistant bacteria, they were able to make it drug sensitive again. Similar observations have been made in other studies, and this also matters in vivo inside the body. For example, metabolites produced by host cells can have a great impact on how pathogens respond to antibiotics. Some metabolites can make pathogens more resistant, and some could make them more sensitive, to antibiotics. So understanding the metabolic environment is very important for predicting or discovering new drugs. And so overall, the picture that people have now are looking at is that, traditionally we thought drugs target a specific enzyme, or antibiotics target a very important pathway in a cell, and by blocking that, it causes cell death. But now we know it's a more nuanced picture where, the metabolism of the bacteria also plays an important role in response to drugs. And also if there are other stress the bacteria is encountering, like oxidative stress or stress by the immune system, all these factor in on how pathogens respond to, drugs. So that's what we want to take into account now when we are trying to build more accurate machine learning models. And so to now build such models, we first generated data on, first try to see if systematically how various drugs respond to various metabolic conditions. And so we took 10 different antibiotics, and then we measured the interactions in LB media, which is the normal media that everyone uses to grow bacteria. And then we changed the media condition and did the same experiment in glucose media condition where there's more glucose and less amino acids. And to our surprise, by changing, making the small change, resulted in a dramatic difference in the drug interactions. You can see here, green is synergy and red is antagonism. And you can see just by switching this, changing the media condition, it's the same bacterial strain. The drug interactions that are highly antagonistic become highly synergistic, and vice versa. So different classes of drugs show like highly divergent outcomes in different media conditions. So that means, the complexity of drug discovery now becomes even more challenging. Like, before earlier I mentioned that the large number of drugs and combinations and dosing, but now we should also take into account the metabolic condition that the cells are growing in. So that results in added complexity when we are discovering new drugs or combinations. So to do that, we developed a new method called Magenta that takes into account the metabolic environment the bacteria is growing in. So Indigo takes in the known drug gene interactions. So Magenta takes in one more input, which is chemogenomic profiles of the metabolic conditions. So these are genes that are, required for growth in a certain metabolic condition like glucose, glycerol, or any other media. So with this additional input, Magenta is now able to predict drug interactions that are more context specific. That means, if it'll predict drug A and B to be synergistic in glucose, antagonistic in hypoxia, or in glycerol. So it makes more context specific drug interaction predictions. So to test Magenta, so we picked a novel media condition that it has not seen before. In this case, we picked growth in glycerol media for E. coli cells, and then we tested whether Magenta can predict drug interactions in this novel condition. And so we also picked new antibiotics that it has not seen before. And so this was once again like a worst case scenario where it's novel drugs and novel conditions. And then we tried to compare Magenta predictions with experiments. And you can see here that once again, just like Indigo, Magenta is able to accurately predict drug interactions in a novel media condition with very high correlation. So synergistic combinations were also confirmed experimentally. So now the next question we wanted to ask was, just like Indigo, can we now use Magenta to make predictions in a novel microbe? Using this orthology approach where we look at the conservation of genes to predict in a different bacteria than it was trained on. So in this case, we picked a bacteria called Acinetobacter, which is one of the most deadliest pathogens in the US hospitals. And then we once again used our Magenta model, found orthologs between E. coli and Acinetobacter, and then we made predictions for this organism. So in this case, we made predictions in 3 different conditions: LB media, growth in glucose, and glycerol. And you can see from the heat maps that the drug interactions change significantly between different media conditions. There's a lot of synergy in glucose, a lot of antagonism in LB media, and it's a mixed bag in glycerol. But once again, Magenta is able to accurately predict how the drug interactions will change in these new media conditions, as shown in the plot on the right, where I'm showing the predicted interaction scores in different media conditions and comparing them with the experimentally measured drug interaction scores. And once again, there was a very strong correlation between the model and predictions. So just like the case where we did earlier in Staph aureus and Mycobacterium tuberculosis, the accuracy is slightly lower than E. coli. And this is expected because you're using data from E. coli and projecting it to predict drug interactions in Acinetobacter. But we're getting this for free again, using E. coli data to make predictions in a different organism. So moving on, we also looked at what genes and pathways were important for these predictions. And we found interesting novel pathways like those in glyoxylate metabolism that were predicted to be involved in drug synergy and antagonism in novel media conditions. And recently people have found that glyoxylate pathway, its activity correlates with antagonism of various antibiotics. For example, it's required for uptake of drugs, and blocking this pathway, would result in decreased uptake, and hence will make drugs more antagonistic. We also seen similar effects in cancer cells. So this might also be a considered phenomena that occurs in other cell types. So in a recent study, we looked at how the metabolic state of cells influences the response to a certain class of drugs called HDAC inhibitors. And what we found was that the potency of HDAC inhibitors dramatically changes depending on the media conditions in which cells were growing in. And we were able to accurately predict the potency of these drugs in, like, for example, glucose conditions or growth in lactate. So these are environments that are similar to what cancer cells might see inside the body. And so this phenomenon seems to be highly conserved across evolution. So we're also trying to scale this method to hundreds of conditions. We're using a combination of metabolic modeling and machine learning to apply this for hundreds of conditions or combinations of metabolic stresses that cells might encounter in vivo. So for the last 5-10 minutes, I want to talk about one final challenge in drug discovery, which is that conditions in the lab are very different in vivo, and I want to talk about how we can better capture this in vivo infection environment using these models. So recently we started collaborating with the Denise Kirshner and Jennifer Lenderman's lab here at UFM, and to combine our machine learning models with pharmacokinetic dynamic models that they have developed. Specifically, we're using this for studying tuberculosis. They have developed a computational model called Grandson that it's a tissue level model where you can accurately simulate the infection site, which is called a granuloma. And what it is that when humans inhale Mycobacterium tuberculosis, the infection pathogen is surrounded by immune cells. And so it forms a ball of cells called the granuloma, and it forms like a tight protective barrier around the pathogen. And so what it means for drug treatment is that many of the drugs might not penetrate the site of the infection or reach the pathogen because it's covered by immune cells like macrophages, T cells. So by forming a barrier, they also form a barrier against antibiotics. So things that we have discovered in vitro, where you add both the drugs together at the same time, they may not reach the infection site at the same time in vivo. They might have different diffusion properties, or they might have different pharmacokinetics, and so that would influence the in vivo drug potency. So to model this, we combined INDIGO predictions with the Grandson model. And so this would help us to predict drug interactions in vivo at the infection site. And what the output of the model looks like is that now we are able to calculate what the concentration of different antibiotics will be at the site of infection, as shown in the heat map on the left. And using that, we can now calculate an in vivo drug interaction score that tells you how synergistic or antagonistic drugs are at a specific infection site or at a specific location inside the body at a given point of time. So that's example I'm showing here on the right. So by doing this, now we can screen multiple regimens and calculate their in vivo interaction score. So here are 4 examples that showed striking differences between in vitro drug interactions and in vivo drug interactions. So the first one is HRZE, a combination that's commonly used for treating tuberculosis. And what I'm showing on the FIC score is the prediction by INDIGO, where it says it's highly synergistic. And we also find that to be a highly potent or synergistic in vivo at the infection site. The reason being that the drugs that are involved in this regimen all reach sufficient concentrations at the infection site. And so they're able to interact synergistically because they all reach the same infection site at the same time, and they're able to achieve sufficient concentrations. In contrast, we have another drug combination, RE, which is also synergistic, but surprisingly is synergistic in vitro, but it's not highly synergistic at the infection site. The reason being that they have very different concentration profiles. Rifampicin is able to diffuse into the infection site at a much better rate than the other drug, EMB, and so they have different concentrations and they're not able to actually interact synergistically in vivo. So similarly, we have other examples where, for example, RM, this drug's combination is predicted to be highly antagonistic by INDIGO, but then at the infection site, they don't actually meet at the same time to be antagonistic, and so they end up being neutral or linear. And so the antagonism predicted by INDIGO does not occur at the infection site. So this is a great way to triage drug combinations from INDIGO, and then now with using pharmacokinetics and dynamics, we can better predict whether this synergy will happen inside the body or not. So now we are able to more accurately forecast clinical trials, and previously we were only able to find drug combinations and not capture dosing, but using GrandSim now we can also differentiate different dosing schedules for drug combinations. For example, the same drug combination, HE, might be dosed differently. So, sometimes one drug is given more frequently than the other. Now we are able to differentiate different dosing schedules. And this helped us to better predict drug regimen efficacy from clinical trials. You can see with a higher correlation. So for the last couple of minutes, I want to talk about one more story where we trying to understand the molecular mechanisms behind how the immune cells eliminate pathogens so that we can design new therapies that work in synergy with the immune system. And so one specific mechanism we want to focus on is on killer T cells. So killer T cells play an important role in removing viral pathogens, cancer cells, and also like different intracellular pathogens like tuberculosis and E. coli. And so we try to understand how killer T cells actually kill these pathogens so that we can use that idea for designing new therapies. And just to briefly talk about the study, we treated different bacteria like E. coli, tuberculosis with what's called as immune enzymes called granzymes that actually kill these pathogens. And then we identified proteins that are being cleaved by these enzymes using proteomics. So we did a simple experiment. We took these three pathogens, treated them with granzyme B, and then we did proteomics to figure out what proteins were being cleaved by granzyme B. And then what we found was that when you overlay that on a cellular map, granzyme B cleaved a lot of enzymes in metabolism, as shown here. Right? A lot of enzymes in TCA cycle, glycolysis were all being cleaved. And one interesting thing we found was that even though a lot of proteins were being cleaved, they were not being done in a random mechanism, in a random way. But this enzyme was going after specific proteins that were highly conserved in these bacteria. So what we found was that most of the granzyme targets were highly conserved in over 300 different organisms. And so that explains why granzyme B is able to kill diverse bacteria like E. coli, tuberculosis, even cancer cells, because it's going after highly conserved metabolic enzymes. And one interesting thing what we found was that by going after a combination of enzymes, it reduces the chance for developing drug resistance. And so when we grew E. coli cells treated with granzyme B for multiple generations, we found that the cells were not able to develop resistance even after like 14 passages. In contrast, when you've done the same experiment with antibiotics, you would have seen that the bacterial growth survival increases continuously over time as the bacteria evolves resistance. But in this case, we could not find any resistance using granzyme B. So to summarize, I talked about 4 different challenges today. One was a large number of combinations that we can tackle using INDIGO, and then using INDIGO orthology, we can apply this for less studied pathogens, and then using MAGENTA, we can apply this in different contexts, metabolic conditions, and then using INDIGO grandson, we can sort of calculate PK/PD pharmacokinetics and calculate drug interactions at the infection site. So, we're sort of moving towards personalized predictions here by developing these tools. We're sort of tackling different challenges due to clinical strains, PK/PD in vivo, and we're also trying to personalize this by understanding host level differences in drug response. That now I would like to conclude and thank various collaborators, like David Sherman's lab, Julie Lieberman's lab for the Granzyme study, Lindemann and Krishna's lab for the Granzyme study, and students in my lab. And thank you all for your attention.
Great. Thanks, Sriram. All right. So if you have any questions, feel free to unmute yourself and ask directly.
This is fairly interesting, Sriram, and we really appreciate it. And, there's so much, future in all this. Have the drug companies picked up on this? I mean, what's the field around you looking like in terms of its evolution and maturation? Where do you see this all going?
So I would say for things like tuberculosis, mostly nonprofits like the Gates Foundation or NIH is the ones that are most interested. But the same idea could be applied, for other diseases. So, like, for example, one of our collaborators in a company, Axela Incorporated, and they are interested in combining different metabolites for treating fatty liver disease and others. So probably not for infections, but, the same ideas could be applied for more commercially interesting diseases. For TB, I think we're working with nonprofits for taking these to the clinic.
Thanks. How much computing is needed to do this?
So I would say probably not a lot. I mean, we use the Great Lakes facility, but it depends on the number of combination screen. For screening 5 million, it takes a while, definitely. But with the Great Lakes cluster, I would say within a few hours, you can make all these predictions.
Interesting.
So I have a question. In your INDIGO system, my impression is that you are considering drugs to be similar if they have similar effects on the molecular level. Do you take into account the structures of the compounds at all, or have you considered doing that in your models?
Yeah, that's a great question. So far, not one thing that we've observed is that some of the errors we make seems to correlate with chemical properties that's not captured in chemogenomics, like how hydrophobic a drug is or how soluble it is. So I think for the next iteration, we are thinking of adding chemical structures like SMILES or other properties in there so that these biophysical effects can also be captured. And so that's definitely on our to-do list.
Nice. Follow-up question on that is, what do you think are the pros and cons of random forests or SVMs versus deep learning approaches for these kinds of problems?
I mean, so ideally, I would like to use a more mechanistic approach. And so with random forest, we can at least get some mechanism. I mean, it's definitely not, white box, but we can at least figure out what genes and pathways are involved. Whereas in deep learning, it's much harder to do that. And so far, we've been trying to use more transparent AI, but if the accuracy goes down, it definitely, like deep learning is the way to go, especially if you're using chemical structures. I think deep learning is much better at capturing those.
Great. Any other questions for Sriram?
do you have any collaborators over in microbiology and immunology? I mean, what's the state of collaboration in terms of this?
So far, our collaborators are at, like UW, like David Sherman's lab have been helping us test for tuberculosis, and I guess Judy Lieberman's lab at Harvard, they have been working on the immunology side. At UFM, we've been collaborating with Denise Kirshner. She's, I guess, in immunology, and so.
Oh, she's great.
Yep. Those are the people we team up with.
Yeah, she's a member of the grad program.
Oh yeah, yep.
Yeah, trained one of our first graduates.
Awesome.
Probably a professor at this point.
All right. Yeah. Well,
All right. Well, if no other questions, thanks again, Sriram, for a great talk, and we'll see you all next week for the next seminar.
Thanks. Great seminar. Thanks so much. And thanks, Josh and Jay.